So we are going to listen to our next inspiration talk. And for that, we are going to welcome Professor Nathaniel Dominy, or Nate. And he's going to introduce a very interesting topic on daylight and human origins. And Nate is the Charles Hansen Professor and Chair of Anthropology at Dartmouth College, and he's also Adjunct Professor of Biological Sciences. His research program revolves around the sensory adaptations, feeding ecology, and evolution of non-human primates. Oh, does that include some of our presidents, maybe? I don't know. But he aims to better inform our understanding of the primate fossil record, especially the key transitions that propelled human evolution. And I'm guessing that when you're in fieldwork, we can find you mostly in tropical Africa, correct? Please, Nate, come up front. The stage is all yours.
Thank you. All right. All right, I just want to thank the organizers for having me here. I'm just thrilled to be here, and I want to thank you, actually. This is my first Daylight Academy conference, and I didn't know what to expect, but you guys are amazing. It was just so thoughtful, so kind, so generous, and I just felt so welcome here, and so I just want to thank you for being such a diverse and multidisciplinary audience. I feel at home. Okay, today I'm going to talk about daylight and human origins. But it's common in anthropology to give a positionality statement. So I just want to give you mine and to say that I have a PhD in anatomy and I have a postdoc in ecology and evolution. So I have a very different background than most of you. I'm in an anthropology department, and I self-identify as a biological anthropologist. Now, I'm a biologist, but we do things a little bit differently in biological anthropology. By the way, we're a rare species. There's only about 2,000 of us globally, so it's a pretty small discipline. Biologists typically have a research question, and then they choose the model organism best suited for that question. Anthropologists do it the other way around. We focus on humans and non-human primates, and we let our model system shape our questions. So that's where I'll be coming at. That's the direction I'll be using today, just to give you a sense of how I think about questions. Now, yesterday was my 50th birthday. Thank you for the party. I really appreciated it. That was a lot of fun. Thank you. And so, you know, that's like a moment to reflect a little bit on like how you got here and the kind of path you took. And so I thought I would just take this moment in this talk to tell you a little bit of a story. And that story begins in the 1990s when I was an undergraduate student. And a paper was published in Nature by a team from UC Santa Barbara. And it was a bombshell paper because they found out that howling monkeys, Americans will say different things. I happened to be working with this species in Costa Rica as an undergrad, and so when this paper came out, it was just really, really surprising. And the story of the paper is that these monkeys have trichromatic color vision. They have 3 opsins expressed in the retina, and they can tell the difference between red and green. Now, other monkeys in Central and South America can see red and green, but it's a weird polymorphism. So 2/3 of females have trichromatic vision, but all males are uniformly red-green colorblind, and 1/3 of females are red-green colorblind. So in this particular genus of monkey, there's a gene duplication event, and all males and females are uniformly trichromatic, like humans, old world monkeys, and apes. And so this was a big, big surprise. And so I just felt compelled at that moment to spend the next 15 or 20 years studying color vision in primates, in part because of the fortuitous event of that paper being published while I was studying that very same species. So this is a recent paper that summarizes the different opsins and whether they're present or not, and it roughly gives spectral sensitivity up here. And so I just want to walk you through it. It's kind of a complicated figure, but it's really impressive. So it's depicting the primate phylogeny. So you have humans and great apes up here, and way down here you have more primitive primates like the lemurs. And what it's showing here is blue. So all primates have a blue opsin pigment expressed. It's autosomal. It's on chromosome 7. In some species, however, you see there's an X here. In some species, there's been mutations to disable that pigment. And so those species are monochromatic. They see in black and white only. And that's a puzzle. So the ancestral condition is to have two present, a middle wavelength opsin and a blue one, but in some primates the blue is lost. And then in other primates, there's a gain. That middle wavelength sensitive opsin, roughly sensitive to green, has duplicated, shifted its spectral sensitivity, and so now you have some that are sensitive to redder colors. And what's interesting, what they've mapped on here is the duplication event. So that big arrow there, it's duplication event there, and it's so advantageous that it sweeps to fixation, and it's preserved in all the descendants of that last common ancestor. So all humans, great apes, and old world monkeys have that duplicated opsin gene, and we're all uniformly trichromatic. Here is the howler monkey. And what makes it so cool is it's a similar duplication event, but it happened independently and convergently with old world monkeys and apes. So it happened twice. And in howler monkeys, they eat a lot of leaves. And so in some ways, it was the most puzzling monkey of all, because the monkey that least deserved trichromatic vision is the one that got it. And so this really created a stir in the discipline, and those of us who study monkeys really were thinking about, well, what are the selective pressures that favor trichromatic vision? And so I decided to do my dissertation work in Uganda in the late 1990s, and I was studying chimpanzees, I was studying monkeys, and I was really interested. They eat different things. Chimpanzees are 90% of their diet is fruit. You get some monkeys that eat mostly leaves, and you get some monkeys that are sort of in between, and they'll eat anything. And so I was just really curious, is there a food target in their environment that they all depend on to different extents that might be red, that could explain why this duplication event was so strongly favored in the last common ancestor of these animals? And I ended up studying fruits and leaves, and I really came down hard on leaves as the answer. And so what I want to paint for you is a picture where we can measure the sensitivity of the receptors in the eye, so we can have a rough sense of how an animal sees color. We can measure the spectral reflectance of objects in their environment, potential reproductive partners, predators, foods, and we can estimate the color that goes into the animal. We can do all of that. But in the same time, in the 90s, you had this citation classic, this really important paper by an evolutionary biologist called John Endler. It was published in Ecological Monographs in 1993. And he says, hang on. You can measure the color of a thing, you can measure the color vision of a thing, but the ambient light conditions matter. And the title of this paper is The Color of Light in Forests and Its Implications. And the implications were profound, both for the evolutionary biology of organisms and their ability to communicate. And that's primarily what he studies. He studies signaling between sexes primarily, but also between species. And he says the perception of a color, whether it's an animal or a flower or a fruit, depends on this interaction between the ambient color of light and the intrinsic color of an object and the ability of that animal to see that color. So light matters. And what was so important about his paper is that he went around to different forests measuring light. And here is what he calls a large gap. Now, the Daylight Academy would recognize that as daylight, right? Oh, by the way, I should say all of these spectra are normalized between 0 and 1, because he doesn't care about absolute quantal flux. He cares about the shape of the spectrum. That's what he wants to compare here. And so this spectrum of daylight is relatively flat, so daylight would be white light. He also talks about woodland shade. And I want to point out that this spectral curve goes down, so it's relatively rich and shorter wavelengths, bluer light, because it's getting a lot of sky, but the sun is being blocked by overhanging vegetation, and so you get a reduction in those longer wavelengths. So shaded light is relatively blue. Now, if the canopy is continuous, if you have constant connectivity between trees, then you get a very different kind of light environment. And the light being filtered through those leaves is extremely enriched in green wavelengths. Right? This is chlorophyll. And so you have a limited amount of blue light, a limited amount of red light, and a lot of green light. And he called that forest shade. Okay. So Endler was moving horizontally, but these same principles apply vertically. And so here's a curve showing white light at the top of the forest. As you get into the shaded part here, you get bluer light, and then as you ultimately get into the understory, it's green light. And you can see the attenuation is rather severe, such that at the forest floor, you're getting about 1% of the light incident on the canopy. I just want to pivot hard here and talk about fire, because 10 years ago, there was an important paper in anthropology about the importance of fire. It was published by Polly Wiesner, an anthropologist at the University of Utah, and she announces her retirement, and then that very same week, she's elected into the National Academy of Sciences, and she's allowed to write her inaugural paper, and she's thinking, what am I going to write about? And she pulls out these analog tapes from the 60s, and she starts, these were recordings of conversations. She starts to codify the type of conversation and quantify what people are saying at different times of the day. And what she found is that at night, seated around a fire, the conversations change from, daytime would be ordinary gossip, like who's sleeping with who and where can we find food, and then at night, 85% of the spoken content was some kind of story, a myth, a fable. And so she talks about this firelight niche, this really important niche, or this firelight space, as probably causing neural entrainment and facilitating communication, the evolution of language, big brain size, big ideas. But in anthropology, we weren't measuring light. And so we decided to start measuring the luminance function of light. Light, by the way, is sufficient for high acuity foveal vision. Some people thought it was too dim. We can measure the spectral properties of firelight. It excites our long wavelength opsons, so our visual system is well suited to being excited by this stimulus. And just a quick shout out to Emanuel Spitzen, he's here somewhere. We're able to take these spectral curves of firelight, run it through his software, and calculate melanopic daytime equivalent functions in lux. And we can see that firelight is a really great light source. It's bright enough to see by, but dim enough that it won't excite melanopsin, and therefore it won't suppress melatonin production. So it's a really perfect kind of artificial light at night. But the other thing we can do is measure near-infrared. There's been a lot of discussion at this conference about the importance of near-infrared and its photometabolic benefits. So here's the emission spectrum of firelight extending past 700. And you can see most of the energy is in the near-infrared, which we perceive as heat. Here's the range that mitochondria absorb that enhances ATP production. And I just want to point out that firelight may have photometabolic benefits. Being seated around a fire for 4 hours, typically, that's what hunter-gatherers do, may confer all kinds of metabolic benefits. So now that we're thinking about near-infrared light, let's revisit Endler's spectral curves and extend them out into the NIR to see what they look like. So this is fieldwork in Uganda in 2022. So Uganda is a small East African country. Here's Uganda here. And this part of Uganda is a large national park called Murchison Falls National Park. This park has a strong rainfall gradient from west to east, such that you have short grass savannas here. By the way, the giraffe is there for scale. You have short grass savannas, longer grasses here with some trees, more dense trees. You have canopy woodlands here, and then you have closed canopy forests with chimpanzees. So here is daylight spectrum from Murchison Falls National Park. You see it closely matches Endler's data, nice and flat here. All right, but when you go out into the infrared, that's what it looks like. All right, so it dips down, that's the oxygen dip right there. All right, but let's look at woodland shade. All right, relatively rich in the shorter wavelengths. All right. Oh, I should point out UV light here is relatively enriched. And then there's the IR spectrum in woodland shade. Okay? So what you really want to think about is this ratio between near-infrared light and UV. All right, here's Endler's forest shade. Here are my data. Matches his data pretty well. By the way, notice Endler looked at this surge in data right here. Right, we get that too. And then here's the near-infrared data. So UV light is practically nil in the forest understory, and then near-infrared light is relatively enriched. So when we think about the ancestral light conditions, when we think about the majority of human evolution and the kind of light we were exposed to, it's right here. Chimpanzees regularly rest in the forest shade, and they regularly rest in the woodland shade. So they're minimizing UV exposure, and they're maximizing near-infrared exposure. And one way to see infrared light, you can't see it, but a proxy for infrared light would be these middle wavelengths, potentially. So if I'm an architect, an interior designer, urban planner and I want to create an ancestral light environment, these are the spectra I would use to try to create that environment. All right, so I just want to circle back to the slide. I'm wrapping up, I promise. Remember, you can lose opsins, you can gain opsins, but one thing you never mess with, natural selection will never get rid of these middle opsins, these green opsins. And I think we can explain that now. Because green light, strongly green light, is your proxy. It's how you predict an optimal light environment. You can't see the UV, you can't see the infrared, but you can see green. And when you see green, that's a really good signal for a nice light environment. All right, we used to measure light in the understory using heavy equipment. You lug it out there, have a portable battery to power everything. And we talked just a few hours ago, we talked about the kinds of equipment you can use to measure near-infrared light. Technology has advanced so much. Now we can scale, now you have something the size of a cell phone, and you can be measuring light from 350 to 950. I mean, it may not be that accurate, but we can start to ask questions like, are chimpanzees preferring particular micro light environments to rest? And they rest in the middle of the day pretty often, and they strongly prefer shade. All right, so I just want to say thank you, huge thank you to you for listening. I want to thank my collaborators in Uganda, Kat Hobater, Funding, Packard Foundation, the Fulbright, and the National Science Foundation, and I'm happy to invite questions.
Thank you, Nate, for this fascinating talk. And we have time for a couple of questions from the audience, if anyone would like to venture and ask commentaries over there. Rona.
Thank you so much. All I'm thinking now, as a hospital designer, if for my doctors and nurses and patients, if I could design a lighting environment where chimpanzees designed for their living and resting space would probably be better, a healing environment and maybe a better working shift environment. So really fascinating, the chimpanzees world, and hopefully we can translate that to the real environment.
Oh, thank you. I hope so.
Any other over there? Thanks.
Richard Weller, Edinburgh. Really interesting stuff. You'll be aware of Nina Jablonska's work, I'm sure, about kind of evolution of skin color and how humans, how we shed our fur and probably then develop dark skin in response to the UV. So that presumably occurs when we move out of the forest onto the plains. And you've then got this very different atmosphere of more UV relative to IR. Is that correct?
So this is Homo erectus. So the thought is, so hominin origins is happening in a woodland. So we develop bipedalism. But by the way, that's the operating definition for being a member of a hominin. The hominin clade is if you walk on 2 feet. So the origin of bipedalism happens in woodland, but Homo, the origin of our genus, happens in the savanna. That's that shift from the woodland shade into true daylight. And I think that's hard. That's a hard shift to make. And so I think you're going to minimize activities in the middle of the day as much as possible. So you're going to rest, preferably in shade. That's the best light to rest in. And then the problem is now you've minimized your social activities. So then I think that's why fire is important, because fire extends your day by an additional 4 hours. And so all those social activities are important at that time. And you get that IR boost. So why not? Fire is really terrific for cooking, socializing, and potentially photometabolism. But you know, all this is all so fringe, right? We need to test these things, but you're the audience to do it. You have all the skills and the methods to do it. We in anthropology, we're like so far behind. But no, those are great points.
I might quickly say Britain today has opened its first open-air intensive care unit on the roof of King's College Hospital in London, and they're going to study health outcomes in patients, just while you bring that up. Yeah.
Oh, that'll be the day when we have fires in hospitals. There's another question over here. Thanks.
Thank you for this wonderful, eye-opening presentation. I'm curious if you have looked at all at aging and differences in behaviors and exposures between little ones, adolescents, adults, and aged populations among primates, or anything there that you can observe?
I haven't personally looked at aging in human populations, natural fertility human populations, or wild primates, but we're finding that because we've been studying chimpanzees since the 1960s, we now have those longitudinal data, and we're finding that chimpanzees are living much longer than we ever thought. That story is being rewritten as we speak.
Perfect. Okay. So I think we can pause for now, but thank you for this presentation because it's eliciting questions, and I hope there's new projects coming in the DLA thanks to you.
I hope so.
So I hope so too, because this is fascinating. As an anthropologist, I'm like admiring you from here. It's perfect bridge to natural sciences, which, as we know, has been the focus of these 2 days.